Hello, so today's topic is about the Yeast 2Hybrid assay. So it is generally used to detect the protein interactions inside a yeast cell nucleus. So it is generally based on the fact that a eukaryotic gene expression is promoted by the transcription factors called the activators. So the Yeast 2Hybrid assay relies on the expression of a reporter gene, that is such as LAGZ or GAP, which is activated by the binding of a particular transcription factor. Okay, and the transcription factor is composed of a DNA binding domain and a activation domain. Okay, so the query protein of interest fused with the binding domain is called the BET, and the protein which will bind or fused with activation domain is called the prey or fish. Okay, so the next is, in order to activate the reporter gene expression, a transcriptional unit must be present at the gene locus, which is only possible if the bait and the prey gene will interact simultaneously. So here is the structure. This is the DNA binding domain and this is the activation domain. This is the DNA double helical structure, and in this direction the transcription is going on. Okay, and as I said, the DNA binding domain and this is the activation domain. So the protein that bind to the DNA binding domain is BAT. Here BAT will attach, and here BAT will attach, and to this prey or fish will attach. Okay, so it consists of two domains. One is DNA binding domain that binds to the regulatory sequence, and another is activation domain that interacts with RNA polymerase enzyme. Okay, and it stimulates the expression of the associated gene. So these two genes can be physically separated from each other, in which case the protein loses its activity. So in order to activate the expression, it has to be in the reunited state. They will be reactivated when the two domains are brought together. Okay, they are brought together, then only the expression will happen. Okay, so the next is, here the proteins that bind to bait, the fish or prey, are fused to the transcription activation domain. Okay, here the DNA binding domain, it binds to bat, and the activation domain, it is binds to fish, or it is also called as prey. Okay, so next is, see, here there is no binding. So the proteins that do not bind to the bait will not activate the transcription of the reporter gene, in this case histidine in this case. Okay, so you can see the proteins that do not bind to the bait will not activate the transcription factor. Here there is no binding, here binding is not happening, that's why it is not activating the transcription of the reporter gene, that is histidine in this case. Okay, but in this case there is binding is occurring. Any protein that binds to the bait will activate the transcription of the reporter gene. Okay, so you can see in this picture the mRNA transcription is occurring. Okay, so this is all about the Yeast 2Hybrid system. So let me summarize this once again. The activation domain of the yeast transcription factor is fused with a polypeptide called the prey. Okay, here it is. This is prey or fish. Okay, and the prey interacts with another polypeptide that is called BAT, which in turn is fused with the DNA binding domain of the same transcription factor. Okay, so the successful transcription will occur when the successful transcription of an essential gene is possible when the bait and the prey will interact, bringing together the DNA binding and the activation domains together, okay, of the transcription factor. So here there is no binding, so there will be no transcription of the reporter gene. But in this case, the binding is there, so there is initiation of the transcription initiation complex, then the expression is achieved. Okay, so this is all about the yeast 2 hybrid system to detect the protein interactions. Thank you. Hope this helpful.